a very warm welcome to everybody to our Science Cafe. And tonight we have two excellent speakers who will be talking about epigenetics. So I would just like to make a small note here that our event today will be recorded and will be uploaded to our YouTube channel for further distribution. And also the views in this presentation and the slides are solely the presenters, and they are not necessarily those of the UMass Medical School. So with that, so our Science Cafe is hosted by the UMass Medical School Postdoc Association, or the UMPA, and as a part of our science outreach activities. And at the end of our the today's session, we'll have a feedback survey, and the link will be posted in the chat box by Alicia. So I would request everybody to fill in the survey and let us know of any suggestions or any topics which you would like us to cover in future, and also your suggestions for improvement. And also we have our YouTube channel, which is the UMass Postdoc Association. And in the past, we have organized other science cafes on some interesting topics like CRISPR and COVID-19 and autism. And in case you've missed those talks, they are all uploaded to our YouTube channel, which you can check out. And also we have our social media handles on Facebook, Instagram, and Twitter. So do follow us there as well. And that brings us to our fourth Science Cafe tonight. So we have two speakers, Dr. Erica Hildebrand and Dr. Melvys Valledor, who will talk about their research on epigenetics and also share their views ranging from chromosome inactivation to regulation of specific genes. So I'm very excited about that. So I'll introduce our first speaker tonight, Dr. Erica Hildebrand. So Erica grew up in Vermont, and she did her bachelor's in biological sciences from the Connecticut College, where she studied gene regulation under Dr. Deborah Eastman. And then she moved to Seattle for her PhD in University of Washington and Fred Hutchinson Cancer Research Center, where she studied centromere epigenetics under Dr. Sue Biggins. Then she joined UMass in the program in systems biology under Dr. Job Decker, and she's pursuing her postdoc there and studying the 3D organization of the genome. And outside the lab, Erica enjoys skiing and hiking. So without much ado, I would welcome Erica on board, and over to you.
Thank you so much. Okay, so I'm going to share my screen now. Okay. All right, hopefully everyone can see that. Okay, so thank you so much for the introduction. I'm really excited to be here today talking to all of you about my research and about epigenetics in general. So yeah, I'm a postdoc in the Dr. Lab in the program in systems biology at UMass Medical School. And so I'm just going to start with an overview of epigenetics in general. So epigenetics, basically epi means upon, above, or in addition. So epigenetics is regulation that occurs in addition to our genes. So these are modifications that occur on top of our chromosomes, on top of our DNA, but they don't actually change the underlying sequence, but they can have really important consequences for various phenotypes or diseases. And so epigenetics can be changed by environmental aspects such as food or your age or various other things, and they interact with your genetics to lead to a specific phenotype. Yeah, so to go into that a little bit further, first I want to talk about genes. So this is what the epigenetics is going to be regulating, basically. And so our genes are contained in our DNA, which we know is in chromosomes in each of our cells. And so you can think of the DNA as the cookbook. It contains all of the different genes that are required to make a cell, to make an organism. And each of these genes, when it is needed to be made in a particular cell, is going to be transcribed or made into another molecule called RNA. So each gene will have a specific RNA or many RNAs that is made of it. And these are sort of like a copy of a recipe. And these, so they can be, they're then be able to be transported out of the nucleus, this is where the DNA is kept, and into the rest of the cell, where they can be then further made into proteins. And proteins are what do most of the work in our cells. So in addition, there are some RNAs that actually are not made into proteins and instead are made into non-coding RNAs. And these have other functions of the cell, some of which are epigenetic, which you're going to hear about more in the second talk. And so just to get back to this metaphor, so we have our RNA, which is the copy of the recipe, and then the protein would be sort of like the finished food. And this is, yeah, what is going to go on to do something in your cell to make a structure or to do a function. So I want to start with a poll question. So just based on what we talked about, do cells in your heart and your brain have the same genome? So just quick first guess, what do you think? I think we're just gonna keep it up for a few seconds. There we go. All right, so yes, most of you agree that cells in your heart and your brain have the same genome, and absolutely, that would be the case. However, there's one genome sequence, but there's many different cell types that have to be made out of that same genome sequence, right? So we know that in your brain, you might have neurons, and in your heart, you might have heart muscle cells. And how this works is that even though we have the same genome in your brain, there are specific genes that are important for making neurons that are going to be on, whereas specific genes that are important for making a heart muscle cell are going to be off. And this is the same in the heart. So how these genes are regulated can be through epigenetics. So this can switch genes on or off in specific cells or specific cell types. And epigenetics can also be heritable either through cell division. So cells in a particular tissue that are going to divide will remain the same type of cells, or they can even be heritable. These changes can even be heritable through generations in specific cases. So there are a few different types of epigenetic regulation. I'm going to go over a few of them today. One of these is called DNA methylation. And so this is when a methyl group, which is just a small chemical, it's CH3, is bound directly to the DNA. So this involves the activity of a protein enzyme that can bind, that can add the methyl groups to the DNA. And this will occur at some regions of your DNA, but not at others. And generally at places where your DNA is methylated, that tends to turn genes off. So another type of histone or of epigenetic regulation is called an histone modification, and these tend to occur. So these occur on histone proteins instead of on the DNA itself. And histone proteins act as a spool which organizes your DNA. So DNA is wrapped around these proteins and then it's packaged into the chromosomes and into the nucleus. But the histones also regulate the DNA. So there are specific signals that can be added to the histone proteins that can turn on genes if they're add a gene that needs to be turned on. And these are called acetylation and methylation. Although I want to note that for methylation, it depends where on the histone. So in this particular region, it might turn it on, but if it's on like the blue, but if it's on the green histone, it might turn off the gene. And then there are also some stop signals that are going to turn genes off that can also be modifications that are added to the histones. And again, one of these is methylation at other sites on the histones, and also one called ubiquitination. And this is a little bit different because methylation and acetylation are both sort of small chemicals, but ubiquitination is actually a whole other protein that gets attached to the histones. So in general, both the type of the modification and the location on the histone that is modified determine the gene activity. And so across your chromosome, there are going to be regions that are modified in one way and regions that are modified in the other way. And this will lead to different ways that your chromatin is that your chromosome is organized, and then different genes will be on or off in those different regions. In addition, there's also 3D organization of your genes or of your chromosomes. And so this is also related to the histone modifications itself. And here I'm just showing you what this looks like in a microscopy image. So this is a cell nucleus. In blue is all of the DNA. So this is all spread out throughout the entire nucleus. In green is the histone modification that turns genes off. And you can see that this is around the outside of the cell nucleus at the periphery. And then in red, with all these little red dots, this is a histone modification that turns genes on. And so you can see they have very different locations in the nucleus. One is around the edge and one is in these little dots in the middle. And in addition, actually regions from different parts of your chromosome that are active will cluster together, and regions that are inactive will cluster together. So this is another way that the epigenetics can be regulated, can regulate gene expression. All right, so now based on this, would you think that the cells in your heart and your brain would have the same epigenetics? We have another short poll. Yeah, okay, you guys, great, you're understanding what I'm saying. So no, they don't, right? We're going to have, they'll have the same types of mechanisms, but the locations of where genes are turned off and on and where these different modifications are going to be located are going to be different depending on whether we're in one type of cell or a different type of cell. Exactly. All right, so what I'm working on in the lab, in the Decker lab, is what controls this 3D organization of these genomic regions that I was just telling you about. So what allows genes that are on to cluster with other genes that are on and genes that are off to cluster with other genes that are off at the periphery. I'm using an experimental system called tissue culture, where cells are grown in a dish in the lab. And instead of using microscopy, I'm using a genomic technique to measure the interactions between different regions of the DNA. And importantly, I've been telling you so far mostly about the 3D structure of interphase, so cells that are not going through cell division, where the DNA is spread out throughout the nucleus. But when cells are dividing, they actually condense their DNA into the X-shaped chromosomes that you might be familiar with. And this is called mitosis or cell division. And then it needs to be reorganized back into the spread out like full nucleus organization as they once they're finished dividing. And so I'm really interested in how they're able to make this huge transition in chromosome folding. And we hypothesized that a protein enzyme called topoisomerase 2 might be involved in this process. So in order to test this, we, using a genomic technique, measured the strength of the 3D clustering, either for genes that are on or genes that are off, based on different epigenetic marks. And so we can see that this clustering, like I was showing in the microscopy, is at a certain level in the control. So when there's no treatment of the cells, so this is cells that have just exited mitosis, just exited cell division, and are now in interphase. But when we get rid of this enzyme by a chemical inhibition, as cells are exiting cell division, we see that now the strength of the 3D clustering is decreased, both at regions where genes are on and regions that genes are off. So this result is showing us that this enzyme called topoisomerase 2 is important for resetting the 3D organization of the genome after cell division. And we're currently studying the mechanism for how this occurs and what the impacts are for cell function. All right, so that's what I wanted to talk about today. Thank you so much for listening. I'm really excited to hear about your questions. And also thank you to Umpa for organizing this.
Yeah, thank you very much, Erica, for this wonderful talk. And I'm sure there are a lot of questions. So everybody, if you have questions, please type them into the chat. And we already have a first one. So I'm Benedict, and I will be hosting or moderating the questions today together with Manish. So what keeps the genes that are on together?
Yeah, so it has, I mean, it's not really known, but one thing is that actually so it's what I'm interested in finding out. But one thing is that the histone modifications themselves may have some sort of intrinsic ability to be attracted to other similar types of histone modifications, and also they bind to specific other proteins that then will bring regions that are similar together. So there's other proteins in the nucleus that are also involved in gene regulation, and so it's thought that that helps to sort of clump together and cluster together the regions that need to be regulated similarly.
So yeah, the next question that we have is, what exactly is the genetic strength measuring technique? You talk in more detail.
Yeah. So I'm using Hi-C, which is a chromosome conformation capture technique. So it's a way of basically you're looking at all pairwise interactions of DNA in the nucleus. So every piece of DNA, what other pieces of DNA it interacts with, and then you sequence all of the interacting pieces, and you can make a map of wherever there's more interactions, you're going to get more sequencing for those regions.
When does this epigenetic switch between the different cell types happen? And who decides which gene is on and which gene is off?
Yeah, no, that's a great question. So this is happening generally during development, so in the embryo, and I think that I don't know the exact timing, but I think that probably different cell types will be differentiated, which is what it's called when they sort of become a specific cell type at different times. And there are specific transcription factors, which are will turn on specific genes, and some of those genes might be involved in more epigenetic regulation. So there's a lot of sort of overlapping regulation and turning genes on that are then going to send the cell into a specific fate or a different fate. Yeah, during development generally.
There's a related question since you mentioned about embryonic development. So what factors, or are they different from the adult? What factors decide which genes are going to turn on and which genes are going to turn off?
So I would say that the overall mechanism is similar in most cell types, whether we're looking at the embryo or later, but there will be specific transcription factors, specific factors that are more important during development than later. And I'm sorry, I don't know a ton of details on this. I'm more studying the regulation in somatic, so in adult cells, and then how it's changing during the cell division. But I think it's a super interesting question. A lot of people are definitely working on that.
And does it differ in X chromosomes?
Yeah, so the X chromosomes are really special, and you're going to hear more about that in Melvys's talk.
Are there chemically created forms of this topoisomerase which act in the same fashion as those enzymes that are naturally made?
Oh, that's a really interesting question. So topoisomerase, what it does, its enzymatic activity is to make breaks in the DNA and then pass one strand of DNA through another. So chemicals that are making breaks in the DNA would have a similar role, except they wouldn't necessarily be then repairing themselves properly. So that's the trick of the topoisomerase, is it can repair properly. So yeah, so I'm not sure, but that's a good question, something I should think about.
And we have another question about another enzyme called helicase. You could probably tell us something about it, and how do helicases influence epigenetics?
Oh yeah, okay. So yeah, so helicases can unwind the double strands of DNA, and they would be really important during DNA replication, which I didn't even talk about. And yes, they definitely would influence epigenetics in that a lot of histone modifications need to be, as the cell is replicating its DNA, so it's going to make another copy of its whole genome, it also needs to replicate the histone modifications so that it knows which genes are supposed to be on or off. So in that way, the histone modifications end up being re-added as the helicase goes through the DNA. So yes.
How long lasting are these effects? So can they be inherited to the next generation?
Yeah, so some definitely can. It depends. So there are some effects. I think there's some studies on a specific diet that can be affect the next generation. There are also epigenetic effects that just affect sort of a particular cell type and then are erased for the next generation. So it depends on the specific one that we're talking about. I think that DNA methylation rather than the histone modifications may last more through different generations.
So a follow-up question then, does our lifestyle affect any of these things?
It can, yeah. So your environment and definitely can affect your epigenetics, and they do tend to sort of change over time as you age or as you're exposed to various different things in your environment. So yes, I don't know that yes. So the answer is yes, but I guess exactly how you can, I don't know that you can really like decide on how it's going to be changed, but over your life it will change.
Um, so in one of the images that you showed about the image where you showed the genes that are turned on and off in the nucleus. So the green signal was where the genes are turned off, and they
Yes.
around the periphery. So is that usual? Like, are the genes that are turned off, they are pushed outside towards
Yeah,
the edge of?
in most cell types, yes, this seems to be. So the genes that are turned off, they have specific histone modifications that will recruit proteins that bind to the nuclear lamina, which is a protein that's around the outside of the nucleus. So it's important for the like the envelope of the nucleus itself. There are so in the most cases, yes, these inactive genes are going to be pulled to the outside. There are some specific cell types that don't have these sort of connecting proteins. And so in those cases, the inactive genes usually end up in the middle of the cell, which is a very different organization. And this is, I think it happens in mouse eye cells in particular, and is thought to maybe have something to do with helping with vision. So it's very interesting.
The next question is, can gravity, for example, in a fertilized egg, be an epigenetic factor? So in birds, deciding maybe, I don't know, probably the sex of the bird, or in reptiles. So does that have anything to do with gravity itself?
Wow, I don't know. That sounds really cool though, and I'm gonna have to go look it up. Does anyone else know about that? I don't know.
I know that in plants, gravity has an impact. I don't know if it's through epigenetics or not. And plants can definitely have a way to sense gravity. And they have a different way to sense it in shoots versus roots. And I bet you
So it might
epigenetics
be.
may be involved, but I'm not 100% sure.
And you know, I think there's something about twin, like the astronaut who has a twin, right? And I think they've looked at his epigenetics and it's different from his twins also. So yeah, possibly.
Right. Yeah, there's another very interesting question. Do viruses or say other bacterial pathogens that may or may not cause diseases, do they modulate your epigenetics?
Yes, absolutely. So bacteria that don't cause diseases, for one thing, I think your sort of microbiota, so the bacteria that are helping you and like living in your gut, can have a big effect on your epigenetics because they might affect your metabolism and what you're able to process from your food, and then that can affect epigenetic changes. And also some viruses can definitely affect epigenetics as well. I don't know a ton about that, but yes, they definitely can.
Okay, so I think if no one else has any questions anymore, at least there's nothing else in the chat, then we just move on to the second talk.
Great,
Thank you, Erica.
You're
Ishani,
still
we can
muted.
hear you.
Oh, sorry. Yeah, so I was saying that thanks, Erica, for a fabulous talk, and really enjoyed it. And the questions were also super insightful. So then moving on to our next speaker, I am not. Okay, yeah. So our next speaker is Dr. Melvys Valledor, and Dr. Melvys, she did her bachelor's in biochemistry at the University of Havana, and she conducted research after that at the Center for Biotechnology and Genetic Engineering in Cuba, where she developed a syphilis diagnostic kit. And then she later worked as a biotech patent examiner at the Cuban Industrial Property Office. And Dr. Melvys obtained her PhD degree from the University of Miami on gene targeting and homologous recombination. And she's currently working as a postdoc in UMass Med in Dr. Jean Lawrence's lab, where she studies the biology of gene silencing, and she's also involved in developing a chromosomal therapy for Down syndrome and also potential epigenetic therapeutic interventions for Alzheimer's disease. So, and outside the lab, she likes winter hikes and music, and she's also involved in projects like fermented food, growing plants, and bringing people together. So, over to you, Melvys.
Thank you. Can you see my slides okay?
Yes,
Yes.
we can.
Yeah. All right. So I would like to thank everybody that came to this event, and I just start by saying that today I will discuss diseases that arise from having an extra chromosome, as you see here. So in this case, you know, you have so many genes from the issue of having an extra chromosome, and how we're going to solve this issue by just completely silencing all the genes that are in that chromosome. As you all know, we all get one set of chromosome from each parent, one set of chromosome from mom and one set of chromosome from dad, but except for the X chromosomes, so we have two genes for each, or we have two copies of each gene. And except for the X chromosomes that are going to be a little different, and we're going to talk about that a little later. So the main point of this talk is that chromosomal disorders are very, very common, and they are the cause of most of the miscarriages in women. And almost like 1% of the babies has genetic disorders. And what we need to think about is that the mutation rates in humans are very, very low, are like probably 1 in a million or below that, while these chromosomal diseases are like almost 1%. So there is a big issue that is like really, really hard to study. And why it's so hard to study is because each chromosome, for example, the X chromosome has like thousands of genes. So it's a complex issue that just to understand what's going on in these diseases, in this with these babies or with these kids, and it's a challenge, but also to treat them is also a bigger challenge. So one of the most representatives of these chromosomal disorders is present in Down syndrome, where they have 3 copies of chromosome 21. And it is the leading cause of genetic intellectual disabilities. And while these children are like really great humans in the sense that they are humane and happy and sociable, and really people love them once you meet them, they also face like huge medical challenges, including leukemia, including congenital heart diseases. So they have to like go through several heart surgeries, and also they develop early on, like when they are like Jones on their 20s or 30s, they start accumulating the Alzheimer plaques, and this is due to a gene that is present in chromosome 21, or at least this is like a big cause. And in fact, this gene was discovered by studying Down syndrome. So there is a good correlation here that is good, like how we learn from diseases by studying what's going wrong in these diseases, and also how we can help them. So I would like to now discuss like how hard it is to treat a single gene disorder, right? So like right now there are all these science, and we are working so hard in developing CRISPR and in developing gene editing tools, right? But how about treating a whole chromosomal disorder that involves like hundreds of genes? So it is really hard. But as I've mentioned before, sex chromosomes are special, and nature already thought about this problem. And sex chromosomes are special because females have 2 X chromosomes, while so this is the way to segregate the sexes. This is the way to like have sexes in you know, in an even way, like 50-50. And so in males we have males they do have only 1 X chromosome, while female has 2 X chromosomes. How do we balance the amount of genes between females and males? The Y chromosome is like really small, so really doesn't account for all the genes that are present in the X chromosome. So nature already invented a solution in which a gene that is produced in the X chromosome are going to be expressed, and it's going to silence all the extra X chromosomes that are going to be in a particular cell, in a given cell. This is going to happen through a mechanism that is epigenetics. And that's why we're going to talk about this today. And in the lab, we are looking at, we look at cells and using the microscope. So this is one of the images that we're going to be seeing later. But and here, what you see in pink is this exist RNA. So this gene is going to be expressed, and it's going to code the chromosome, and it's going to silence all those genes, and it's going to take that chromosome and put it in the corner and put it in the closet, and all those genes are going to be shut off, and so there is not going to be expression from those genes. And this is the way that the cells have to deal with this extra chromosome or this excess of information. So here in the top, I'm showing you this chromosome because we're used to see these chromosomes when the cells are divided, that they have this particular, they're individual, they have this particular shape. But during this stage, the genes are off. So if we want to study like gene expression, we cannot look at this stage on the cell. We usually look at the interface that Erica discussed later when the chromosomes kind of expand. But as these chromosomes expand, they don't just take over the whole nucleus. They kind of poof a little bit, and they each one occupy a little territory. So when we look at them, they look like a little ball. So later you're going to see exist and the chromosome, and they're going to be like this round shape and not this other shape. So here we start with the first poll question, and the question is, what does exist gene encode? Does it encode a protein that silences a chromosome, or an RNA that compacts a chromosome? Oh, great. All right. So I saw that still some people think that exist is a code for a protein, but exist is one of these genes that it never gets to make a protein. It only produces an RNA, and that RNA has a function that is like to silence a whole chromosome. In fact, most of our genome only produce RNA, and it never goes to protein. And the function of that those RNA is like one of the things that we're studying in the Lawrence lab. So exist is an RNA, and and let's see how we do this. And but it originates from a gene, and what Dr. Lawrence and the laboratory did a couple of years ago was to realize that this mechanism to silence a whole chromosome exists in nature. We all have it in our cells, in our body. And so they took this gene and they put it in cells coming from a Down syndrome patient that was now like a stem cell, was made into a stem cell. For that, they had to use a genetic engineering technique, zinc fingers nucleases in this case, that will cut the DNA right at the specific place that they were they wanted to send the gene. In this case, they wanted to send it to chromosome 21, right? So they were successful after a long time, and they got it into the chromosome 21. And so after that, and then they made this gene inducible. So after you add this molecule DOX, that activates expression of this gene, you could see they saw that the same thing that happened to chromosome X happens in chromosome 21. So this gene exists, was also painting the chromosome 21 and stick it in the corner, and also silencing gene expression. And here I'm going to show you an example of a gene that gets shut down. So I told you before that these kids, that the people with Down syndrome develop these plaques in their brain and they get Alzheimer's. And there is a gene that has been that is the one that produces plaques. It's like the gene that produces a protein that is those plaques that accumulate. So having too much of these genes produce these plaques in the brain. And so this gene is called APP, amyloid precursor protein. And while this exists, is able to silence the whole chromosomes with about 400 genes. Now I'm going to show you a microscope image of just expression of that gene. So here is this gene called APP coming out from as an RNA from those genes. So this is amazing that we can see this. Like here is a nucleus from a cell, and here we can label the RNA exactly coming from that gene, so in that precise location. And what happened, and here in red is X6, but it's just the gene. So but what happened when we induce the expression of X6? Well, X6 again codes the whole chromosome, and what happened to the genes in that chromosome? So in this territory here under the red, it will be all those genes in that chromosome. So here in the image below is exactly that nucleus without the red signal to see what's going on under the red signal. And as we can see, there is no expression of any of this gene APP from that region. So this was amazing. When they saw this, it was like, oh my God, we can silence the whole chromosome. So with this system, it's incredible because now we can also study what is happening in Down syndrome, what is happening in these cells, what are the consequences of having an extra chromosome. But also we can study how X6 works in human cells. This is in part because this process of X6 expression only happens very early in the embryos, and of course it's very hard to study human embryos. Almost difficult, almost impossible. So we know very little about how this gene X6 works, and also we wanted to study how exactly this X6 gene is silenced in chromosome 21 for therapeutics purposes. So I would like to show you now how X6 can compact a chromosome, and here is again in a nucleus, and here is how it looks an active chromosome that is expanded while it's expressing genes, right? It's expanded, and the transcription machinery can get into that DNA sequence and express genes and produce genes. But once that X6 gets there, it compacts that whole chromosome, and this is how it looks in the X chromosome. But in the chromosome 21 already we also saw the same thing. So here is the cell, a trisomy cell, we have X6 integrated into that gene into that chromosome, and when we express X6, we can see that the chromosome is also compacted. So here it is. I also wanted to show you a little bit about the Epigenetics mechanism that Erica discussed before. Here in this cartoon, what we're seeing is, like in red, is the X6 RNA. That is a molecule that has these structures. And because of these structures, is able to recruit or interact proteins in the nucleus, and those proteins may have or has different activities, including the modification of like the histones that Erica was mentioning before. So in the panel here, I'm showing how we see cells at the microscope. We use fluorescence, so we label molecules that we fluoresce in different at different wavelengths, and here we're seeing this mark that is active is acetylation of the histones. So it's one of these marks, so this is a stem cell. So usually in stem cells, chromatin is open and there is a lot of expression from a lot of genes, and so this active mark is present throughout the cell. But what happened under X6? Here is a mark in red of X6. What happened under X6? That mark is evicted. Those histones are no longer acetylated. To the contrary, X6 also recruits other marks that are known to be repressive marks. In this case, the methylation that Erica mentioned before. So in this stem cell, so there is little methylation because, again, the genes are active. But what happens when X6 codes the whole this chromosome? Well, it brings, here is X6 in red, it brings this histone modification that is called, that is a methylation on the H3K27. So there is much more. We just concluded a study in how X6 work in trisomy 21, and we are working on we're trying to publish that study, but I'm gonna stop here because of time. But you are all welcome to look for that. So with that, I hope I have convinced you that with X6 we have kind of leveled the playing field in in solving chromosomal disorders, because now the problem is limited to like sending X's to that chromosome. So editing one gene into the chromosome that has a problem. And so it is still a challenge, but it seems more doable than taking care of hundreds of genes. And with that, I would love to thank the Lawrence Lab, especially Jeannie Lawrence, which it is the mind behind all this amazing science, and all the people in the lab. I also would like to thank Lisa that helped me a lot to put this presentation together, and a bunch of friends also that help me to get this presentation clear. I would like to thank the funding sources from the lab, and also some funding from NIH that fund my work. I would like to thank people from Down syndrome that has donated cells and people that make this work possible. And I have another quiz. Let's see this time. How does Exist work? Through epigenetic gene silencing or through changes in DNA sequence? All right. Okay, so I guess the point got across, and most of the people got that it's through epigenetics. And yes, we do need to insert Exist. That's a little confusing. So Exist needs to be expressed from the chromosome that is going to be silenced. So that's the part that is a little confusing, but its mechanism of action is through epigenetics. With this, I will take any questions.
Right. So thank you, Melvys. That was such an informative talk. So yeah, so the questions have started trickling in. The first question that we have is, do other organisms also regulate their extra X chromosome with Exist, or is it unique only to humans?
Oh no, it's mammals. All the mammals do this, but nature have come out with different ways to compensate the sexes. For example, let me think if I get this right. So some other organisms will increase the expression of the X chromosome in males, and some of them will dampen both of the X chromosomes to make up of one. And then there are other organisms that will do things like with temperature, they will define the sexes, but that will probably make more sex, more females or more, dependent of the temperature. So probably that's hard to control.
But is this mechanism, or is this kind of a system exist only for X chromosomes? Why did nature not devise it for the other chromosomes?
Could you repeat the question?
Why did nature not devise this for the other chromosomes? Why just X chromosome?
Well, the other chromosome, well, because it's good, I guess it's good to have two genes, because they're you have, I mean, to have two copies of each gene in case that something goes wrong with one gene, and also it provides diversity. So I guess it's a good thing to have two copies of a gene, but also we need a way to come out with different sexes, right?
Okay, thank you.
Yeah, we have another question from the audience. So how does expression of Exist on one chromosome target only the extra chromosome in, for example, trisomy 21, and not the other two, even though they are genetically identical?
Right. So this is also amazing. We don't know exactly how it does it, but Exist only silence the chromosomes that is expressed from. So it doesn't affect other chromosomes. So normally in nature, it will only affect the X chromosomes in females, one of them, and for that there is a counting mechanism. And once that exists is being produced from that X, that is going to be remembered through cell division, and that chromosome is going to stay silent as the cells divide. But yeah, in the chromosome 21, it's only gonna be expressed from that chromosome that Exist is inserted on. So the other two chromosomes are safe. and the other chromosomes in the cells are also safe.
So I have a related question. So
Okay,
how
thank you.
how does the cell decide which X chromosome to silence?
Oh yeah, that is another also very interesting topic. There is a counting mechanism, and there are a bunch. It's called the X inactivation center. So it's like 100 megabase of information genes there, and there is a bunch of long non-coding RNA again that doesn't go to produce a protein, but they work right there, and that regulate. Um, they are going to regulate X6 expression. Um, that I'm not an expert in this topic, but I also know that we don't know everything about it. So it's a topic that is under investigation.
Right.
In the case of chromosome 21, we only put that gene without the whole regulatory system. So it's gonna only be produced in the chromosome from that gene specifically.
What are the implications for silencing the third chromosome of a fetus known to have trisomy 21, for example, in a prenatal treatment?
What are the implications?
Yeah, or what would be the implications in the future?
Yeah, so we are not thinking. I mean, I am not thinking that far away and about treating like prenatal, but that would be great if it could be done. If the family wants, because it will avoid a lot of the disease complications that these kids have. But in the Lawrence lab, there are several projects involved in like the onset of the problems in Down syndrome, and what are the changes between a Down syndrome or of cell and a cell that only have the two chromosomes 21, and so with that we're also learning when the problems arise, and also will inform us when do we really need to treat those cells.
Okay, so is cell division a necessary requirement for chromosome inactivation using exist, or can this be used in non-dividing cells as well?
Well, that's also interesting. It's a very good question. In the past, it was thought that that only stem cells could do this, and we don't really know. But a study from a student in the lab, he did show that it was possible to get excess action in neuroprecursor cells. Those cells are dividing, but he also did some experiments that show that excess could be expressed and produced in cells that were not dividing. Yeah, so we don't really target like completely differentiated tissues, and that's also something that we would like to test.
Right.
But maybe a follow-up question. So would that be possible theoretically? So similar to the mRNA vaccines that are used at the moment to get like the RNA into the cell and then, affect the cell itself?
Yeah, so that's also a very cool question, and it was something that we wanted to try, but so we would it is interesting because exists only silence the chromosome that is expressed from, so we don't really know exactly what are the requirements there. If that will be possible, that's something that will need to be tested. If you send exist specifically to a place, and if it will silence genes there, it might be that it requires a lot of excess molecules to be able to do the job, but we really don't know exactly that if that will work.
Can you do these kind of experiments to test if exist can be used to treat diseases in some animal models? Are there any models known which have duplication of chromosomes?
Well, yeah, the Lawrence lab is working in Kushali, which is here. I think she's like trying really hard to work on this, and previous investigators in the lab also. And it has been like a challenge to work in to create to do a model. So right now there are some Down syndrome models in mice. It has been like really hard also because in mice they don't have the chromosome, they don't have exactly chromosome 21 as we have. So it has been like a difficult thing to study, but there are a couple of models now, and that is the work of Kuchali, and we will hear from her soon.
Right, looking forward to that.
Yeah.
Also probably a related question. So then can you also target other chromosomal disorders?
Yes.
Not just trisomy 21? Yeah.
Yes, so something that is very important that I don't know if I really transmit is that this gene doesn't work. It's not specific to a DNA sequence, so it works from any chromosome that is integrated from. So it works in the chromosome that is the gene is part of. So if it is in chromosome 13, it can go and it could express and will code that chromosome and silence that chromosome. If it is in chromosome 18, then that single chromosome will be silenced by exist.
Right. Thank you, Melvys. I think that's all the questions we have. And thank you for patiently answering to all the questions. So I'll hand it back over to Ishani to sort of wrap up today's event.
Yeah, sure. So it was a very exciting event today, and thank you, Melvys, for a very interesting talk. And we had some great questions, and thanks to both the speakers for addressing them so well, and also sharing your research with us. And just a final reminder, I'll quickly share my screen. So just a final reminder to please fill out the survey Alicia posted in chat box, and we would like to hear from you about the topics that you would like us to discuss in future. And also, if you're interested to share your research with us, do reach out to umpa@umassmed.edu. And with that, so I mean, hopefully I mean, till now, like, our science cafe events have been virtual, but we hope to host these events someday at an actual cafe where we want to meet everybody in person. So but till then, we'll have to keep it virtual. So thanks again, everyone, for joining in, and do keep a lookout for our next science cafe event. So bye. See you everyone at our next event.